So, very good afternoon to all of you, and welcome to this Advanced Fluorescence Microscopy and Image Processing course. So, today is our first live demonstration, and let me introduce myself. I am the course instructor, Dr. Gunjan Mehta from IIT Hyderabad, and today I have my PhD students with me. She is Akriti Kumari, and she will also help us for this live demonstration. Okay, so before we begin, I would like to give you some time in case if you have any questions from whatever the classes that you have attended. So by now, I think you must have gone through the 4 lectures. So if you have any questions from any of the lectures, feel free to ask me the question now. You can just raise your hand and start asking if you have any question from any of the previous classes. I hope I'm audible, right? Just say yes or no.
Yes, sir, you're audible.
Okay, all good. Okay, so then I will start with the demonstration. In the meantime, if you have any question, just unmute yourself and start asking. Okay, and you can also type your question in the chat box, but the chat box questions I will look at the end. But during the demonstration, if you have any question, just unmute yourself. Okay, so yeah, now I will take the camera to the microscope. Okay. Just trying to. Okay, so all of you can see this microscope. So see, this is a standard fluorescence microscope, okay, and it is from the manufacturer known as Leica. So this is a German company, and there are 4 main manufacturers in the microscopic field. So Nikon, Olympus, Zeiss, and Leica. So this microscope is built by the German company Leica, and I will just show you from all the 4 sides and how it looks like, and later I will explain you the individual components of this microscope. So generally all these sophisticated microscopes are generally connected with a computer, and they are totally automated. Means once I put my sample inside, I do not have to touch the microscope, and entire things I can control through the software. Okay, so let me explain you each of the component of this microscope. So let us start from here. So here you can see this kind of a black box. Okay, so this black box is actually the light source. Okay, and it has, as you can see on the display, there are 8 LED lights in this black box. Okay, there are 8 different colors starting from violet to green, yellow, red, and red. Okay, so there are 8 different LEDs are present in this light box. Now, how the light is transported from this box to the back end port of the microscope. Okay, so you can see one cable. This is known as the optical fiber cable. Okay, so this optical fiber cable takes the light from this box, and it takes the light to the back end port of the microscope, from where the light enters to the microscope. Okay, now this microscope can operate in 2 modes. Okay, so in one of the next class, you will see that there are several modes of illumination. The most common one is known as the epifluorescence mode or the epi illumination, and The one other type of illumination mode is known as the TIRF mode, which is known as total internal reflection fluorescence. Okay, TIRF mode. So this microscope is equipped with two illumination modes: the bright field or the wide field illumination mode or the epi illumination mode, and the TIRF illumination. Okay. Now for both this mode, it needs different light sources. For the epifluorescence mode, it will use this light source where there are 8 different color LED lights in this box, and it will use that light for the epi illumination. Okay, but when we have to use this microscope under the TIRF mode, at that time you can see another light source is placed here. This box is known as the laser box. Okay, so this box has 4 different wavelength lasers. Okay. So when we have to use this microscope under the TIRF mode, we have to use these lasers as a light source. But when we have to use this microscope as an epi illumination mode, then it will use this LEDs as a light source. So I hope now you have a clear understanding about the light sources. So the same microscope can be equipped with 2 different light sources. One is the LED light source and other one is the laser light source. And depending on your application, you have to switch on the appropriate light source. Now coming to the camera. Okay, so here you can see this is the camera which is connected to the left hand side port of the microscope. Okay, and this is Prime 95B S CMOS camera. Okay, this is currently one of the latest camera in the imaging field, and it is S CMOS camera with 95% quantum efficiency. Now, the camera that you people use in your mobile phone is also mostly the S CMOS camera. Okay, and this is also an S CMOS camera. But there's a huge difference between both the camera types. Your mobile camera is a color camera which can capture the color images. Okay, but generally in microscopy, a monochromatic camera is used. So monochromatic camera means this camera cannot see colors. It can just capture the image as a black and white and grayscale image. Okay, this camera cannot see colors. And why the monochromatic cameras are preferred in microscopy? Because here we want to quantify the fluorescence intensities, right? So let's say if I take the image of a sample in 3 different colors like red, green, and blue, then I have to quantify the fluorescence intensity in these 3 colors, right? And for that, if I produce the RGB image, that is a merged image of all these 3 colors, okay, then I cannot individually quantify that fluorescence intensity. So instead of capturing the multicolor or the RGB image, it is always advantageous to capture the individual channels separately. So that's why the monochromatic cameras are generally preferred in microscopy, so that you will capture the red color fluorescence separately, green color fluorescence separately, and the blue color fluorescence separately. Tentatively, just for your information, these kind of cameras are super expensive. The cost of this camera itself is 25 lakhs Indian rupees. Okay, now why they are so expensive? Because inside this box, there are some electronics components involved to maintain the temperature of the camera chip at minus 30 degrees. Okay, and why this minus 30 degree temperature is required? Your mobile phone camera does not operate at minus 30 degrees, but this microscopic camera operates at minus 30 degrees because the camera itself has some inbuilt noise. Okay, so if you operate this camera at room temperature, it will show some noise in the image. But if you operate this camera chip at minus 30 degrees, then that inherent noise is reduced. Okay, so to reduce the noise in the image, this camera operates at minus 30 degrees. That's why they are highly sophisticated. They are so expensive. Now I will show you now this microscope. So in your third lecture, you must have studied there are 2 configurations of the microscope: the upright microscope and the inverted microscope, right? So this is kind of an inverted microscope. Okay, this is not an upright microscope. And why it is inverted, that I will show you. So see, here in this microscope, this is the sample, and below that you can see the objective lenses. Okay. So you can see the objective lenses are at the bottom of the microscope. Okay. See, in the inverted microscope, objective lenses are at the bottom and they will see the sample from the bottom. Whereas in upright microscope, these objectives are placed somewhere here, so the objective will look at the sample from the top. Whereas in inverted microscope, the objective will see the sample from the bottom. So if I put the sample here like this, then my sample will be observed from bottom. Okay, because this is an inverted microscope. Generally, in biological applications, the inverted microscope is preferred because we have to see the live cells. Okay, and live cells are generally kept in the liquid culture media. So see, this is the imaging chamber in which generally the cells are grown. Okay, now if this imaging chamber has the liquid media, and if I want to see the cells from top, then I cannot go really close to the sample because my objective is touch to the liquid. Okay, whereas from the bottom, this imaging chamber has a very thin cover slip kind of a bottom. Okay, so because of that thin cover slip, the objective lens can go really close to the sample, and the shorter working distance objective, like when you want to image something at a higher magnification, like 100x or 60x. Okay, at that time you need a very short working distance. So with a very short working distance, you can actually see your samples. Okay, and that's why generally for biological applications, where you want to image live cells in liquid culture, for those kind of applications, people prefer generally the inverted microscope over the upright microscope. But for if your application is more on the material science side, then it is better to go for the upright microscope. Okay. Now I will show you two modes of illumination. So this microscope or any other fluorescence microscope is generally equipped with two types of illumination: transmitted light illumination and the fluorescent light illumination. Okay. So here I will show you how the transmitted light illumination works. Yeah. So on this and switch on the transmitted light. Okay. Okay. So see, this is an inverted microscope. So the light source for the transmitted light is placed here. So there is one bulb inside this black box. Okay, that is kind of a regular bulb, or I would say not very expensive bulb, because it will just give you yellow or white color light. Okay. So for transmitted light, the light source is here. The light passes all the way through this tube, and this is the condenser. Now this is an inverted microscope. So condenser is at this position. Okay. And from the condenser, the light falls onto the objective lens. Okay. And when you put the sample in between, so this is the sample position, you will put your sample here. So in transmitted light microscopy, the light will pass through the sample and the transmitted light will go into the objective. Objective will collect that transmitted light and it will pass on the transmitted light either to the eyepiece, so you can see the cells here, or it will pass on the light to the camera depending on your need. If you want to capture the image on your computer, you will pass the transmitted light to the camera so that the image can be captured. But let's say you want to see the cells under the microscope by looking at the eyepiece, then you will transmit that light to the eyepiece so that you can see the cells in through eyepiece. Okay. So this is a transmitted light path. I am again showing you in transmitted light path. The light source is at the top. Now this light source is different than this LED light source. Okay, this LED light source is used for the fluorescence illumination, whereas this light source is used for the transmitted light. Means you just want to see the cell morphology or cell size okay, under the bright field illumination, then you will use this transmitted light. So this light source is here, that bulb is on, that light is passes through this route. This is the condenser. From condenser, the light will go to the sample, and the transmitted light from the sample will be collected by the objective lens here, and the objective lens will transmit the light either to the camera or to the eyepiece. Now, how the fluorescence light paths work? Okay, so for fluorescence mode or the epi illumination mode, the light source is here. Okay, the LED light source, and it has 8 different colors LED. So, which 8 different color LEDs are there that I will show you. As I mentioned that this is a completely automated microscope, so everything is controlled from the software. So this is the software interface, and here you can see there are different colored LEDs are available. You can see that 390 nanometer, 440 nanometer, 475 nanometer, 510 nanometer, 555, 575, 635, and 747 nanometer. So these 8 LED lights are present in that box, and from the software, I can manually, I just switch on the light, and I can also set the intensity. Like currently you can see the 475 nanometer light is on, and the intensity I have set to, let's say, 70% or 60%. Okay. So this is how I can switch on or switch off the LED light in that box from the software, and I can set the intensity of those lights. So what is the light I will select from here? That light will be switched on in this box. That light will pass through this optical fiber cable. It will go to the backend port of the microscope. Okay, and from the backend port, it will come all the way through here, and from here, it will go up through this, and there is a kind of a fluorescent filter placed here. So the light will pass through the fluorescent filter, and depending on what fluorescent filter I have selected, it will pass the specific wavelength of light will pass from here, and it will go to the objective. Okay, so I will show you how it looks like. So yeah, switch on that 475 nanometer light. Okay, so can you see that blue color light? So that is the 475 nanometer light that we have switched on from the software. So in software, you can see the 475 nanometer light is on with 100% intensity, and that light you can see at the object. So here, see, the light is coming from the object. Okay, it is not coming from top. It is coming like the light, the excitation light itself comes from the object. So, okay, so the excitation light itself comes to the objective, and then if you put the sample here, let's suppose your sample is here. So this light will illuminate your sample, and the sample will reflect some light. So that reflected light will go back to the objective. It will again pass through the emission filters here, and that the emitted light will be collected by the camera. Okay. Generally, under the fluorescence mode, it is not recommended to see directly in the eyepiece because we use some light in the UV wavelength also. So it is not a good idea to observe your fluorescence sample directly through eyepiece. But instead, if your microscope is connected to the camera and the software, it's better to observe the fluorescence images on the software rather than directly by your naked eyes. Okay. So again, I'm just showing you the light path for the fluorescence light. So fluorescent light is coming from the backend port of the microscope from here. Then here is the filter wheel. Okay, I will open this thing to show you how the filter tube look like. And let me just do it right away. So I open this. So here you can see different filter tubes. See, this is a filter tube for DAPI. This is a filter tube for GFP. This is a filter tube for Texas Red. So different filter tubes are installed, and I will just take out one particular filter tube. Let's say take out the GFP filter. So see, this is how this filter tube looks like. Okay. Now this filter tube has 3 filters or mirrors inside. So this is the this is filter number 1. Okay. So this filter tube is placed in the light path like this. So the excitation light goes this way. Then there is a dichroic mirror here. Okay, you can see one mirror at the 45-degree angle in this tube. So the excitation light goes this way. It will be reflected by the dichroic mirror, and it will go to the sample. Okay, it will go to the sample, and the reflected light from the sample will come back to this probe, and it will pass through the dichroic, and it will be emitted from it will pass through the emission filter here, and then it will go to the camera. So this is how the filter tube is placed in the retina. I'm again explaining you again. So see, it is placed like this, and this is your light source like this. So the excitation light goes this way from, and it will pass from the excitation filter. Then there is a dichroic mirror here, so it will reflect the excitation light. So it will the excitation light will take 90-degree turn, and it will this way. It will go to the objective to illuminate the sample. Then the reflected light from the objective, it will come back here, and it will pass through the dichroic mirror. It will pass through the emission filter, and then it will go to the camera. So this is how this filter tube works, and it is placed in the light path. And again, this filter tube also operates automatically by the software kit. So you do not have to do this thing manually. Moving the filter manually, you can just select different button, and also I put that filter tube back to its position. Okay, and this is known as the filter turret, because this is kind of a circular frame in which total 6 or 7 different colored filters are installed. And from the software, you can see these are my different filters for selection. So now the GFP filter is selected. But if I want to change the filter to M cherry, I just have to select it from here. This is M cherry, and you can see how it rotates the filter wheel. So see, now I am changing the filter from the software, and it will rotate this filter wheel, and the appropriate filter will be placed into the light path. Okay. So this is how the filters selection works automatically from the software. You just have to select the filter. Accordingly, that filter will be rotated. It will bring appropriate filter in the light path. So this is about the fluorescence illumination. Okay. Now see, this kind of microscopes are also equipped with the live cell imaging chamber. So this is a live cell imaging chamber. So basically it's a small chamber that you can see. Okay. And in this small chamber, this wire is connected, some tubes are connected. So this wire is to maintain the temperature. So let's say for live cell imaging experiments, we want to maintain 37-degree temperature. So we have to adjust that temperature in this box, and it will maintain that temperature. Also, through this tubing, it will maintain the level of carbon dioxide or any other gases you want to maintain. Or even if you want to maintain some humidity, that can also be maintained through this tubing and the water system rotation. Okay. So this is known as a live cell imaging chamber. So when we have to image the live cells, we put this chamber on the top of the stage like this. Okay. So only at the time of live cell imaging application, we have to put this chamber here. Otherwise, this chamber is generally lying outside. Fixed cell imaging, you can directly put your chamber on the stage like this. Now I'll show some regular controls of this microscope. So you can see there is a fine and coarse adjustment knob. So just like the knob present in the school microscope, through this knob you can move the stage up and down. So this is the coarse adjustment knob, and this is the fine adjustment knob. And this microscope is also equipped with the joystick. This is known as the joystick. The purpose is the same. Through this joystick, you can move the stage up and down using this knob, and X and Y using this knob.
Excuse me, sir. Actually, we are not getting clearly voice, sir.
You are not getting clearly?
Voice, sir. Your voice is vibrating, sir. And
Oh,
echoed.
Oh, is it? Okay. Let me just
Thank
check.
you, sir.
Some disturbance from the live stream. Is it clear now?
Yeah, sir.
Okay, okay. So I will just proceed, but let me know in case again you find any difficulty in hearing. Okay. Okay, so we already discussed about this live cell imaging chamber and the two different light paths, the fluorescent light path and the transmitted light path. And what else? Okay, so I will just show you the main panel of the microscope. So see, this is the main panel, which is a touch screen panel. Okay, and as I mentioned that this is an automated microscope, so just by touch screen you can select different options and appropriate optics will come into the position. So see here, you can select different modes of imaging. So see, this microscope can take images in bright field, fluorescent mode, fluorescence plus phase contrast mode, DIC, that is DIC plus polarizer, and fluorescence plus DIC. Okay, so these are different modes in which mode you want to image that you have to decide and select. So let's say you just want to take the bright field image, then you select this mode, so only the transmitted light will turn on. If you want to take an image in the fluorescence mode, you have to select this option, so the fluorescent light will turn on. Okay. And also you can select the filters from here also. So you can select the filters from software also. And from here also you can select the filters, whether you want to see GFP or M-cherry. Also the objective lenses you can change from here. So see, there are 6 objective lenses installed in this system. 10x, 20x, 40x, 63x, 63x, 100x. Now you must be wondering that why there are two 63x objective lenses, right? But can you see this extra mark here, the oil droplet, right? So that indicates that this 63x is the oil immersion objective, whereas this 63x is the air objective. Okay, so when you use this objective, you have to put a drop of oil on your sample, whereas by using this objective, there is no need to put an oil. And I think in one of the class, you must have studied that why the oil immersion objectives gives you better resolution. Okay, because by putting the oil between the sample and the objective, the more of the reflected light will go into the objective, and that will provide better resolved image. So depending on your application, you have to select the objective lenses. And this option is there to adjust the X, Y, and Z position of the cell stage. So all these things you can control through this control panel. And this microscope currently it is in the open position, so that you can load the sample, you can put the slide on your stage. But once the slide is there on the stage and you really want to start imaging, you have to close this chamber. Why this kind of closed box is given? To avoid the exposure of external light to the sample, because the stray light or the external light can also create some disturbance or noise in your image. So that's why they have provided this kind of a dark box here, so that while using the microscope or while imaging, your sample is not exposed to the external light. And here you can see this is the software interface. Today I won't show you more about the software interface, but in the next live demonstration on next Friday, I will show you how to use this software to capture image of 5D or 4D images for live and pixels. So see in the software, you can see there are multiple dimensions to select. Whether you want to take only image in XY or XYZ, also you want to add time. So depending on your requirement, you have to select this mode. So suppose you want to take a Z stack image, you have to add this Z. So software will show you some options to select the Z stacks, that how many Z stacks you want and what should be the step size, and all these things you can see, number of steps, the Z step size. Okay. So when you click this Z, then only it will show this option. If you will add the time, this T, then it will add this time interval panel. So in the time interval panel, you have to define that what time interval you want to take the time lapse movie. You want to take the image at every, let's say 5 minutes, every 10 minutes, and for how long? For 1 hour, 2 hours. So those things you have to define here. So once you start the imaging, the software will automatically take the images in X, Y, Z time. If you set multi-position, then it will go to different positions on the slide and capture the images. So I think that's it for today's demonstration, and I'm ready to take the questions if you have any. So any questions from today's demonstration or anything that I taught in last 4 lectures? Okay, so if there is no questions, then we'll just go offline, and feel free to ask questions. You can post your questions in the discussion boxes, or you can even feel free to write me an email also. Okay. Okay, so thank you so much for attending the demonstration. And our next demonstration is scheduled on next Friday at the same time, between 5 to 5:30 or 5 to 6 p.m. Okay. See you in the next week. And in the meantime, you will also go through the other 2 lectures, lecture number 5 and 6. Okay. Okay. See you and have a nice weekend.
Thank you, sir.
Thank you, sir. Thank you very